in silico vaccine. So let's start with the introduction. We all know that the first vaccine was introduced by British physician Edward Jenner in 1796, and he used the cowpox viral vaccinia to confer protection against smallpox. It was just an accident, and the first vaccine was accidentally discovered. Vaccines can be whole organism. It could be live attenuated or heat killed. It could be a protein, a small peptides. It could be DNA based, mRNA based, epitope based, multi-antigen vaccine, or it could be multi-epitope based. So there are different categories for vaccines. In last 2 decades, the reverse vaccinology strategy has been extensively exploited, especially during pandemic. We all are aware that during COVID-19, everyone was looking for a very good, reliable vaccine against SARS-CoV-2. And during that time, lots of research groups are involved in this reverse vaccinology. So all over the world, different research groups explore this vaccinology approach. And the first vaccine that was discovered by this reverse vaccinology approach was Neisseria meningitidis. And after that, there are lots of pathogens like Plasmodium falciparum, Leishmania, Zika, and other pathogens were explored for this reverse vaccinology approach. So why reverse vaccinology approach? Reverse vaccinology is a very cost-effective approach because most of the time we start with the whole pathogen. But in case of reverse vaccinology, we can just focus on proteins that can be further used for vaccine development. There is a whole flowchart we will discuss in our later slides, but it's a very cost-effective and fast process. And because it includes computational approaches, immunoinformatic tools, it's very fast process. Like you can get a vaccine using reverse vaccinology, it could be 2 or 3 days. And after this, there is a new term that is known as immunoinformatics. It's another approach that includes different prediction tools, like it could be B cell epitope prediction tool, T cell epitope prediction tool, antigen prediction tools, and there are lots of tools we are going to discuss in today's session. So let's start with the introduction part of this session. What happens when an antigen or a foreign particle enters inside our body? For example, there is a virus that enter inside our body. What happens? This foreign particle or virus is going to interact with the cell receptor. The host APC is the host cell that could be human or animal. So here I'm going to talk about human vaccine and human immunology. So I will consider this APC is from human cells. So what happens? This cell contains some cell receptors that helps this bacteria or virus interacts and starts phagocytosis. After this phagocytosis, this bacterial or pathogenic particle enter inside this cell, and there is a proteolysis activity start that degrade this whole microbe into small. Okay, Siva. Any questions, Siva? You can unmute and you can ask.
What is APC?
APC is antigen presenting cell, any cell inside our body that interacts with the bacterial cell or pathogen, any pathogen, it could be virus or bacteria, that is considered as APC.
Okay, ma'am. Thank you.
Okay, welcome. So this cell degrade the whole bacteria into small peptide particles or epitope parts. It could be class 1 epitope or it could be class 2 epitope. So MHC refers as major histocompatibility class 1 and class 2. So what happens if the epitopes break into class 1, it presented on the surface, and if it's MHC class 2, it also present on the surface. What happens if there is MHC class 1 and MHC class 2? Both are going to activate our immune cells. In case of CD8 T cell, it activates cytotoxicity and induces memory cells. And what happens when CD4 cell activates? It also induces some T cell immune response, that is Th1 associated and Th2 associated. TH1 induces interleukin 2 and IFN gamma cytokines, and TH2 cells induce interleukins 4, 5, and 13. Besides these, B cell also activates. B cell immune response activates some memory cells for future purpose, and it also induces some plasma cells that induce antibodies in our body. This is the first interaction of any antigen in our body. So this is the overall immunology of our human body. Okay. What happens if it interacts with our innate immune system? So this TLR is refers for toll-like receptor. On the APC, there is toll-like receptors also. So whenever a pathogen enters inside our body, sometimes it interacts with the toll-like receptors. And on the surface of pathogens, the part that is interacting to this toll-like receptor is known as pathogen-associated molecular patterns, and the short form is PAMs. So this PAMs interact with toll-like receptor, and it also helps to increase T cell immune response, and it also induces some interleukins, interferons that also helps to reduce the parasite or pathogenic load. So why we are using vaccination term? What is the benefits for vaccination? What happens whenever a person gets vaccinated? So normally I have told you that whenever a foreign particle enter inside our body induce some kind of immunity. So why we need vaccine? Vaccine is developed to protect ourselves from some kind of pathogenic infection. It could be bacteria or it could be virus. So to prevent ourselves from any kind of disease, we use vaccination. So what is inside the vaccine? We all are vaccinated during our early first year. Like whenever a child take a birth, there are different vaccination process. So that is to prevent them from childhood disease and for future disease. Nowadays, I hope everyone has aware with the SARS-CoV-2 and they have some kind of vaccine. So during the vaccination, what happens? This vaccine may contain live pathogen. It could be killed pathogen. It could be protein of that pathogen. It could be mRNA type vaccine or any particle that is coming from the bacteria or pathogen or virus. If it enters inside our body, it activates our immune cells and it generates some kind of memories. So whenever next time the real pathogen enter inside our body will be ready to fight against that real pathogen. So this vaccine is just to induce immune cells that can prevent us from pathogens. So what happens whenever we prepare a vaccine, we also induce some kind of adjuvants. I hope my pointer is visible to everyone.
Yes, ma'am.
Okay. So if this vaccine antigen, we use vaccine antigens. So with vaccine antigens, we also use some adjuvants. So what happens? Sometimes the vaccine antigen are not very capable to induce the immune system inside our body. So we use adjuvants. These adjuvants act like a catalyst, you can say. So it helps to induce the higher level of immunity inside our body. So we always, or most of the time, we use adjuvants every time we do vaccination. So these adjuvants could be protein based, lipid based, or any other kind of adjuvants. So these vaccine antigen and adjuvant start with immune cell, it activates. So there are receptors already, as I told you that there are receptors present on human cells. So they receive it and they degrade these antigens and present the peptides on the surface of MHC class 2 or MHC class 1. So if you see here, you can see T cell receptors. So these T cell receptors interact with these MHC class 2 or MHC class 1 presented epitopes, and then they start their immune system. So what happens if CD4 cell activates? It helps to activate B cell also. So when B cell activates, it could be activated through T cell activation, or if we are using B cell epitopes in our vaccine, it could be directly interact with the B cell receptor, and it directly induce or proliferates these B cells. So what happens when this B cell activates or proliferates? Sometimes it produces plasma, different, like you can say it's differentiate into plasma cells. Sorry. So it produces antibody, and at the same time, they also generate some memory B cell for future use. So in future, if you are going to, like, for example, you have taken a SARS-CoV-2 or COVID-19 vaccine. So next time, whenever you are getting infection with SARS-CoV-2, these B cell are going to activate, and it's going to protect you from that SARS-CoV-2. And if you come to this CD8 T cell, when it activates, it also generates some effective cells that starts cytotoxicity, and it also generates some memory cells. All these happen inside our bone marrow. So this is the whole story of a vaccination. So if you're going to study about vaccination, or you are planning to develop some kind of vaccine against a specific disease, you have to understand the pathophysiology of that disease. So here I'm showing you an example for Plasmodium. Plasmodium is a pathogen that can cause malaria. So if you see, this is the pathway of, or you can say the life cycle of mosquito. So what happens whenever a female mosquito bites and take a blood meal, it infects or transfer this plasmodium inside our body. So it transfers from blood, and from blood it enters inside our liver, and it generates different kinds of, like, there is different stage for this pathogen. So this is a very complicated pathogen. So if we start with this liver cell, after, so it's very, it develop very fast. Here it take only 24 to 48 hours. After that, it goes to infect some other liver cells, and it changes its form. Like, it enters, whenever it enters inside our body, it's in form of sporozoites. But when it enters inside the liver cell or hepatic cells, it changes into schizoids. This is another form of this parasite. So every time a pathogen changes during its life cycle, expression of protein on the surface of this pathogen is changes. So there are lots of proteins that express only at the stage of sporozoite. And when the pathogen changes into schizoid form, the protein expressed on the surface of that pathogen will change. So what we have to decide, which stage of vaccine we are going to develop. So after this schizoid stage, it ruptures and it goes to blood. So here you see the mosquito transfer this parasite in the liver cell, and after liver cell, it goes to our blood again, and in blood, the RBC, it infected with, and after that, it matures and rupture, releases the schizoids and some prophozoites, and it could transfer into mosquito during second meal, or whenever a new mosquito take a blood meal. So with blood meal, these gametocytes enter inside the mosquito. So this is a whole cycle, it's ongoing. So this part is from mosquito, and this part is from host. So for Plasmodium, there are two different hosts, mosquito and human. So in malaria, scientists are targeting human as well as mosquito for development of vaccine. So most of the time, they target the blood stage. But in blood stage, it's very difficult to destroy these pathogens because these are inside the blood cells. And it's in liver cells, it's very difficult to identify, like it's very, very slow growth. Like it take only 24 to 48 to grow inside these hepatic cells. And when it grows, it will not come to direct contact with our immune cells. So it's very difficult to develop a vaccine for this. The second problem is we have to identify what are the proteins that are expressing on the surface during liver stage and during blood stage. So if you're going to develop a vaccine, you have to study the whole pathophysiology of your parasite. This is just example of Plasmodium. Because it has two different hosts, it's very complicated. But if you're going to study only a single host, like if you're using Zika virus or any other virus, in that case, if there is only one host, human host, still you have to identify what kind of pathophysiology is going on in that specific pathogen. So here, it's a simplified form. You can see you can develop a transmission blocking vaccine or anti-infection vaccine or blood stage vaccine. So every stage you can see there is sporozoite, merozoite, and gametocytes. In each stage, the proteins expressed on the surface of Plasmodium are different. So if you are going or you are planning to develop a pre-exotic stage vaccine, you can use some different proteins. And if you're using asexual blood stage vaccine, you have to target different proteins. So how to identify the vaccine target or the antigenic proteins? If you are new, you want to develop some vaccine candidate, you can go to CADD pathway. You simply write your disease name or pathogen name, and you will see this kind of structure. So what's going on here? You see, there are different species of Plasmodium. When it entered inside this human cell, what happened? You see CSP and TRAP. CSP is pathogenic protein. TRAP is also a pathogenic protein. And these are interacting with human receptors. So this is one part. This is second part. You see the malaria merozoite. So it's a merozoite stage. So here there are lots of antigen. You can see MSP, MA, AMA1, DBP, EBA, RBP. So these are all antigenic proteins that are interacting with human cells. So if you can generate a good antigen or a good uh, vaccine candidate using these proteins, you will have a promising vaccine. If you go further, you see there are toll-like receptor signaling pathway also going on here. So if you see there is TLR9 and TLR2 and 4 are interacting with these kind of proteins. So yes, you can also target this group of protein for vaccine development. And again, if you see there are lots of interleukins that are involved in immunosuppression and some other immune system, like it activates macrophage. So macrophage can destroy that pathogen. And also it also activates the NK cells. So this is just an example for Plasmodium. You can explore the CAG for any other pathogen. So this is just how it works. Like if a Plasmodium falciparum comes inside a host cell, it interacts with the TLR26 or TLR21, TLR4, TLR5. Like there are different kind of toll-like receptors. So whenever these antigens interact with toll-like receptors, there is a signaling starts and it activates the next molecule, and it at the end of this signaling, it can induce some cytokines. It could be TN alpha, interleukin 6, interleukin 12, or some other. This is a list of bioinformatics tools that you can use for vaccine development. These are all free online available tools you can explore. So if we start with a whole pathogen, so there is possibility that that pathogen has more than hundreds of protein. For example, I'm showing you one example. For example, you have one pathogen and you have 208 proteins for that pathogen. What happens when you use some tools for antigen prediction? For example, if you're using Vaxigen, it's a tool that you can use for antigen prediction. You will have 69 antigens. You see, the number of protein decreased. After that, when you use TMHMM tool for transmembrane helix prediction, so we use this tool just to minimize the number of transmembrane helical containing proteins. So what happens if a protein has lots of transmembrane helix? It will not exposed on the surface and it will not be available to our proteins, our immune system to recognize those proteins. So it could be a difficult part for epitope prediction and for our immune system also. So during vaccine prediction or vaccine development, we always prefer those antigens that has at least 0 or 1 transmembrane helix. After that, you can also go for signal peptide prediction. In signal peptide prediction, you can see there is only 15 antigenic proteins that has signal peptides. And most of the time, we try to develop a vaccine that are either membrane or extracellular proteins because it could be easily uh, exposed to our immune system. That's why we always prefer to membrane and extracellular proteins. So you see, the 208 proteins comes to 8. These are highly probable antigenic proteins that we can use for our vaccine development. So this is a simple flowchart. So this is a paper for monkeypox virus. It's a newly emerged virus. If you are interested to develop some vaccine candidates or you want to predict some novel antigens, you can read this paper. So how we predict our vaccine? It's a multi-stage process. So what happens every time we start our vaccine development? We try to identify antigenic proteins. Once we have our antigenic proteins, we have to predict epitopes. So we can predict B cell epitopes or we can predict MHC class 1 or class 2 epitopes. So what are antigens, epitopes, and antibodies? So the part here you can see the small parts that's in continuation. These are amino acids. Okay, so these amino acids are interacting with the antibody. Therefore, we say this is epitope in case of B cell epitopes. So the continuation of amino acids that are interacting with antibody, we say this is a continuous linear epitope, or we can say these are B cell linear epitope. In this case, you see there are amino acids, but they are not in continuation. What happens? An antigens, when it comes to tertiary structure folding, there is possibility that some amino acids interact with antibody and some are not. So there are only 4 amino acids. You can see the small round particles here. These are interacting with the antibody and these are not continuing. If you see here, 20, 32, 47, 50, these are the amino acid residues of this particular antigen that are interacting with this antibody. So in this case, we say this is a discontinuous B cell epitope. So this is clear, like linear, what are linear B cell epitope and what are discontinuous epitope. So every time we predict B cell epitope, it could be linear or it could be discontinuous. So the prediction tools available for discontinuous, there are lots of tools are there. but the accuracy is low. But here in linear epitopes, there are lots of tools and the accuracy is very good. It's difficult to identify discontinuous epitopes because it changes based on the protein configuration, and whatever it holds, it changes. So it's difficult to predict discontinuous epitopes. And similarly, in case of MHC class 1 and class 2 epitopes, we can predict MHC class 1 epitopes for MHC class 1, and we can also predict for MHC class 2. So this is a small protocol. Okay, there is a question that how does the life cycle of a pathogen influence antigen selection in silico vaccine? Very good question. So if you are planning to develop, okay, can I take a pause here? And I want to go back to answer this question. So here, if you are predicting an antigen for a plasmodium, for example, suppose you got 10 antigens, 2 from this region, 3 from this region, and 5 from this region. If you are planning to develop a blood stage vaccine, you have to pick the antigens that are expressed during merozoite stage or asexual blood stage. If you are going to pick the antigens that are expressing during mosquito stage, it will not benefit us very like if a human is infected. So for transmission blocking vaccine, we use those proteins that express during mosquito stage. Like when the plasmodium is inside the mosquito, there are different protein expression. For example, these proteins. But when it enters inside our human host body, the protein expressed on the surface of this plasmodium are different. So if you want to develop a vaccine that can inhibit the growth of that pathogen and that can provide protection during early stage of malaria, you have to select the proteins that express during pre-erythrocytic stage. And if you want to develop some blood stage vaccine, you have to select only those proteins that express this stage. For example, if you are planning to develop an early stage vaccine and you are selecting these proteins, it will not be beneficial because your body has immunity against this. They don't have immunity against this. So it's a waste. Okay, I hope I'm clear.
Yes, ma'am. Ma'am, how much time does our body take like to give immune response towards the vaccine?
So it depends.
Depends on
Mostly,
host
like,
immune
yes,
individual
it depends on
immune.
host individuals. Yes,
Okay.
most of the time it start with first week, and most of the time it takes 21 days. So it depends.
Okay, like whenever we have vaccine shots, immediately we get fever. So
Yes,
can
that's
we?
our immune system responding.
So
The response
immediately our immune system will
started, it's
respond.
activated. Yes.
Okay, okay, ma'am. Thank you.
So this is a flow chart. If you are interested to generate a novel vaccine, what happens? We just predict antigen. After antigen prediction, we have to predict some B cell epitopes. And using these B cell epitopes, you can also predict class T cells, or you can directly predict from the antigen. So here you have two options. Either you can predict your T cell or MHC class 1 or class 2 epitopes using these linear B cell, or you can directly predict from antigen. After that, you have to select because whenever you enter your antigen, you will get thousands of epitopes. So you have to screen out highly probable and highly potential T cell epitopes. And we call it epitope ensemble preparation. After this, you have to design your vaccine. For vaccine designing, we use adjuvants and linkers. We use adjuvants to increase the immune response or the immune induction of our body to support our vaccine. And linkers we use to join these adjuvants with linker epitopes. So linkers are some small peptides, like it could be 2 amino acids, 5 amino acids, or sometimes 8 or 12 amino acids. So what they do, they just help to join the epitopes. Like if you're using 5 epitopes, you can use these linkers. So what happens if you don't use linkers? If you are not using linkers, what happens when it enters inside our body? There is some proteolytic enzyme in our body that degrade these proteins. So if we use linker, it will chop or it will cut at the linker sites. It will not cut at your epitope site. So we want our epitope safe so that it can easily express on the APC cells or exposed on the APC cells. That's why we use linker. Another benefit of linkers, it helps to fold our vaccine in a proper way. So there are lots of benefits of using linkers. After that designing, we have to perform some molecular docking. So you can use TLR2 or TLR4. Receptor in your study, you have to study the pathophysiology of your particular pathogen. For example, if you're using Zika virus and it interacts with TLR7, you should not use TLR2 and TLR4 in your study. So how you will know? You have to study the pathophysiology of that particular pathogen. After that, you have to perform some structural and functional properties predictions. And there are some immune simulation tools available on the, like it's freely available. You can use immune simulation like CIMM. It's a software or tool you can use. It's freely available. After that, you can also explore some antibody predictions. Once you got your good molecular docking scores, you can go for molecular dynamic simulation. And after that, you have to perform some codon optimization for your recombinant protein development. So these are all in silico work that you can perform. For each stage, I have written some tools, like what kind of tools you are going to use for each stage. So so there are two stage, like there are two, you can say if you are going to work on a particular pathogen, there are two possibilities. Like there are lots of research already going on. So you can find some already known antigens, or you can work with computational tools and you can develop some novel antigens. So for already known antigens, you can search IDB, Violin, okay? So these are the database that can give you the already known antigens. But if you are interested to finding some novel antigens, for example, there is new emerging another pathogen. For example, SARS-CoV-2 was novel at that time. Now monkeypox is novel. Past year, like last year, there is lots of monkeypox cases reported from different countries. So what happens if you have got monkeypox and epidemic? So you can predict some novel antigen by using these tools like Signal P, Vaxgen, KMHM, and PredGPI. After that, you have again two options. Like there are already known epitopes. You can use these known epitopes, or you can predict new epitopes. So for known experimentally reported epitopes, you can explore IDB database. If you go to the IDB database, you can easily find B cell epitopes, what kind of cells they are using, what kind of antibodies they are inducing. Every information is available on this IDB database. And if you're going to predict some novel epitopes, you can use IDB, NetMXCPan, and there are lots of online available tools you can explore. After this, you can generate your vaccine candidate and you can predict secondary, tertiary structure, You can also modify your tertiary structure using mod refiner tool, and you can also use Swiss model or your so for Swiss model, you have to be careful with Swiss model if you're generating a novel vaccine, because Swiss model only predicts for already known proteins. If you're using, like, if you're developing a novel vaccine, Swiss model is not a very good um, software for your protein. I suggest you should use ITASCR for your design vaccine. After that, you can check the Ramachandran plot. If it's below 90, you can use mod refiner. And SimImSim, this is the tool, this is the link that you can use for immune examination. And for, okay, now you have your uh, designed vaccine, you have to go for experimental validation. For experimental validation, you need to express your protein. So for protein expression, you need some kind of plasmid that can express those protein. For that, we use JCAT software and SnapGene software. So once you are ready with your protein, you can download SnapGene. It's freely available only for 1 month. So be careful if you are going to download this SnapGene, because it will only available free for 1 month. So once you develop your vaccine, then only download this SnapGene. You will get free access for 1 month, and then you can work with your protein expression. And your docking and dissemination you can perform with Autodoc, HeadDoc, Pymol, and Gromac. So any question, anyone? Okay. If there is no question, I will move forward.
Ma'am, in Plasmodium, there are different type of strains, but only Plasmodium falciparum is taken as most virulent one. So, are there any kind of secreted proteins or expressed proteins on its surface?
Yes. So every species has different, like, Plasmodium is highly variable pathogen. Like, even in a single host, if you isolate that Plasmodium, you will find different proteins. Even if you have a same protein, the sequence will be different. It's highly variable pathogen. So you have to choose which stage and which pathogen. Like, it could be Plasmodium falciparum and vivax. So in humans, most of the time, Plasmodium falciparum and vivax are predominant. So you can select anyone. And once you select your antigen, please go through the blast P and check if there are some conserved sequence or not. Because even after falciparum, there are different strains, 3D7, and there are lots of strains for Plasmodium falciparum only. So you have to perform a blast P, conserved sequence analysis before going for vaccine development. Okay?
Ma'am, this Plasmodium also infect pigs, ma'am.
Yes.
Okay, there is a different strain of Plasmodium that affects in like it infects pigs.
Yes. There are different, that could be different. Like, it also affects mouse. Like, Plasmodium also affects mouse. Like, it affects different animals. It also affects monkey. So
Hmm.
in different, if you are going to develop human vaccine, you have to select only human based strain.
But I am interested, like how do animals will b- produce immunity towards that kind of uh, infection?
Okay, so your research is based on animal based vaccine development.
No, ma'am, no, ma'am. Like, are there any other type of immunity, like any other kind of pathway? Like humans and animals also share similar type of metabolism or similar type of reactions, immune reactions. So, are there any kind of immune reactions that it will trigger in that animals? I just want to know, is there any other kind of pathway that will trigger them to get natural immunity towards infections?
So both have similar kind of epitope. If you use vaccines, they will generate antibodies. They will induce some kind of interleukin. So these are similar things. Like there is no very like particular thing that you have to target. Okay, this is going to work or not.
Okay, ma'am.
I will show you IDV, and I think you will get to understand what I'm trying to say. I will show you some example in IDV. Okay.
Actually, ma'am, can I ask a question, ma'am?
Yes, please.
Yes, actually, when she is asking question that's also I am having some query. So actually, naturally, our body is means our immune system react according the antigen, whatever the antigen enter in our body, accordingly it will produce the antibody. So from
Okay.
that concern, can we means explore the drug or a vaccination? To that aspect means I think the previous one this such type of question is arising that we are designing the means the vaccine depending on the stage or where it will be actually at, but we can also means inversely we can also do this by this way, but means in if it is the body is reacting or body is acting on that particular antigen, so how the body will react, so such kind of proteins or antibodies they are producing. So that phenomena we can use for designing the vaccine.
Yes. Yes. That's why I told about the pathophysiology. If it's human, you should study about the pathophysiology of human. If it's mouse, you should study the pathophysiology of mouse. Every
Okay.
time you are designing any vaccine for cow or sheep, you have to study the pathophysiology of that particular host or pathogen. Because there are lots of pathogens that are common in human host and animal host. And they are jumping from animals to human. So like monkeypox, it was initially infecting only monkeys, but it jumped to the humans now. So now we have to work on those antigens that can activate the immune system in humans. So we can't rely on the monkeys' immune system that are generating or that are inducing those antigens. For humans, it could be different. So we have to develop vaccine accordingly, host base as well as pathogen base, and also the life cycle. What are the stages?
Like, ma'am, in the discovery of smallpox, there was a cow man
Yes,
who noticed that there is a cowpox virus. Like cows also get some kind of infections. So Edward Jenner, he has taken some uh, kind of liquid from that pustules, and then he transferred those pustules. Like he transferred the cowpox uh, secretions into humans to get immunity against smallpox virus.
yes.
So that is the same analogy I am trying to connect here. So suppose if animals also share same kind of reactions or similar type of infections, can we take consider it as an vaccine?
Okay. So this is what Pasteur did. But most of the time, if your immunity is strong, you can survive. But if your immunity is low, there are lots of like more than 100 children died at that time. I hope you remember the history of that cowpox. So why that happens? Because they are not using the heat killed or the dose of that specific cowpox first.
Yes, yes, ma'am.
So the pathogen that are infecting cow and the pathogen that are infecting human, they are different, or sometimes they could be same. After evolving, the most of the virus and bacteria, they are jumping from animals to human. So there are some kind of changes. So if you are using the cowpox virus, it could be better for humans, but again, the efficacy could be reduced because if that pathogen evolve to enter or jump into the humans, there could be some changes, like mutational changes. So in that case, you have to select the pathogen that are infecting or affecting the human host. You can't rely on that animal-based pathogen sequence or any protein. For example, there is a monkeypox virus and there is no vaccine for monkeypox. So people are using uh, another vaccine that is not for monkeypox, but that is similar, quite similar, like they are showing some cross reactivity, but still they are not very effective. For example, smallpox. If you can take smallpox vaccine, you will be protected from monkeypox. So there is some similarity. On the basis of that, that antibody can also help to protect against that kind of pathogen. So in one word, if you want to select the pathogen that are infecting animals, you should try to generate those proteins that are from human host, because these pathogens are evolving and there are a lot of variations. If you have performed some kind of phylogenetic analysis, suppose you have a pathogen, you have 5 sequence of a pathogen from animal, 5 pathogenic sequence from human, and you perform some kind of phylogenetic analysis or multiple sequence alignment, you can easily see that in humans there is different pattern and in animals there is different pattern of protein for the specific pathogen. So if there is a single amino acid change, your immune system cannot respond. I hope you all know about the SARS-CoV-2. There are different variants. Omicron variant. So initially there is COVID-19 and then second year there is Omicron. In Omicron there is different variations. So if you are trying to develop a vaccine with high efficacy, you should go with the protein sequence that is affecting humans, not to the animals. Is it clear? Hello? Any other query? Yes, we have to work on host pathogen also, because there are different kind of receptors that present on the human cells, and when it interacts with the pathogen, there is different kind of immune system ongoing in our body. So we have to also study that host pathogen interaction. If you go to the CADD pathway database, you can easily understand. I will show you the CADD pathway and you will understand it. I will simplify with the example of SARS-CoV-2 because Plasmodium is little complicated. So when I show you the demo class, you will understand. So what happens if you predict epitope using here I used ABC pred, and you will get lots of epitopes. For example, you see this is the linear B cell epitopes. You will get start position and you will get the scores, you will get some antigenic scores. Based on this, you can minimize the number of epitopes because you can't use all these epitopes and generate your vaccine. And if you predict MHC class 1, you will get some kind of different alleles, your epitopes, and the population coverage. And similarly in class 1 MC class 2, you will get some epitopes, predicted population coverage, and different alleles. So after that, you have to combine all those predicted epitopes in a vaccine form. What happens? We always use a linker, EAAK or any other linker, prevent the cleavage from or to predict the cutting of this area. This is just to protect or prevent our protein or vaccine for designing. If you see the red part, this is the adjuvant part. It could be protein. I said the adjuvant could be protein or it could be lipid or any other. Sometimes we use alum for adjuvant, but here we are using protein. So I'm showing the red color in. So adjuvants are in red. Then again a linker. Then these black parts, these are epitopes, and the green regions, these are linkers. So this is just a B cell epitope example. You can generate multiple, like you can use the different combinations. For example, you see CTB, cholera toxin B, this is adjuvant. Epitope 1, epitope together, epitope 3 and 4, 5 and 6. And here you see, I jumbled these sequence. You see, E9 and 8, E9 and E10 comes before like E5 and E6. So you can also generate this kind of jumble structure. Why these kind of jumble structures? What happens when we develop tertiary structure or 3D structure? There is possibility that these epitopes are not exposed outside the surface. So during that study, we can find if we generate multiple jumble structure, there is possibility that we have multiple structures that has multiple epitopes exposed on the surface. Sometimes if you change the sequence of these epitope, you will get some highly stable structure. And we all know that there are some kind of structural changes or configurational changes whenever you change the epitope or peptide location from one position to another. So whenever you are going to design a vaccine, always prepare some different sets of epitope. Here I'm showing 4 different. So you can generate. If you are not able to generate these kind of structure or sequences, you can use VaxiDB. VaxiDB is a software. You can predict. You just have to provide your epitopes, and it will give you these kind of multiple combinations, possible combinations you will get from there. So after that linear structure, we have to predict secondary and tertiary structure. This is the output of this Cypret tool. And this time, I suggest don't use Swiss model for novel predicted vaccine. Always use ITACCR. Because here, if you use Swiss model, you will get only partial part of your designed vaccine. You will not get the full length sequence of your protein or full length structure of your designed vaccine. So always use ITSCR for full length structure of your protein or your vaccine candidate. After that, you can predict the tertiary structure using the same. And if there is some structure issue, you can use mod refiner. After using mod refiner, you can dock your vaccine with a toll-like receptor. Here I'm using TLR2 as an example. This is the most important part I want to discuss. Here you see, this is the toll-like receptor. And most of the time, I have seen the students just interact randomly anywhere. So we don't have to use that kind of method. We always have to use the specific particular region that is surface exposed and that is specifically made for, or you can say the binding site location. Wherever the binding site available for this TLR2 or 4 or any other toll-like receptors, always use those binding sites for docking of your designed vaccine. If suppose your vaccine is going to bind in this part, if this is inside the cell membrane, the whole docking is useless. Okay, so always use binding sites of toll-like receptor for docking purpose. After docking, you have to prepare a table like this, where you can use a control as a C1. Any control you can use. So what kind of control you can use? You can download a TLR structure from PDB database, where you can easily find some complex form of protein with TLR2. Download that protein and use that as a control. Here, in case of TLR2, there is EFEMP1 protein that is interacting with TLR2 receptor, and you can easily find that structure using 2ZC7A. And if you download that structure, you can easily find that how many binding sites are available in this region. And using that binding sites, you can use your binding site and docking study. If you don't have any PDB structure, you can also go for literature search on PubChem or NCBI. Sorry, PubChem, not PubChem, it's NCBI. Go to the NCBI and you start your literature search, you will easily find the binding. So always go for experimental validated binding sites, not the docking or computational identified binding sites. Always go for experimentally reported binding sites. If you don't have that in literature, download PDF file, open the PyMol, or if you have Discovery Studio, open that PDF file. You can easily find what are the atoms involved in the hydrogen bonds. You can easily find in Discovery Studio. So by this, you can write that scores. You can use Autodoc or HeadDoc and write the score. I hope you are all familiar with the docking score. So minimum the score, better the binding. You all know that. After that, you can also study or you can analyze the interacting residues of TLR2 and your receptors. Or you can also include that, okay, these are the binding sites. For example, in here you see tyrosine at 297 position interacting with lysine 4. So lysine 4 is your vaccine amino acid residue, and tyrosine 297 is the amino acid residue of your receptor. So always prepare this kind of table. It's a very good table for overall analysis of your study. After that, you can generate this kind of vaccine recombinant DNA for your vaccine expression. So here, the black region is the plasmid. The red part, these are the inserted cDNA. So we all know that protein, we can translate the protein into DNA back. What we tell that it's a cDNA. So those cDNA can insert inside the plasmid. After that, you have to generate a sequence to identify that if these kind of cDNA are expressing the same kind of protein or the vaccine that you have designed or not. So for that, you have to select the cell that you are going to express. Like if you are going to use E. coli, you have to select E. coli for JCAT. JCAT is a tool that convert your vaccine protein sequence into cDNA. And once you get the similar protein, you can go with this plasmid. You can order on like GenScript. There is a GenScript company that can provide you this kind of plasmid, and you can express your protein in any cells, E. coli or mammalian cells, whatever you want. But always make sure that you can get an open reading frame, this yellow line, after this recombinant DNA development. If you are not getting this kind of ORF reading, your recombinant DNA is not going to express your protein. So always make sure that you have a complete open reading frame for your design vaccine. So this is an example. When you open your SnapGene software, here you can see you can easily use any kind of protein or any kind of cDNA here. So here you see vector. When you click on vector, you can get lots of options and you can upload your file from here. When you come to the insert, you click here, you will get options for your cDNA. Just upload the file and you will see here, this is the vector, this is your cDNA protein, and this is the product. So when it shows this kind of thing or this kind of pop-up here, ready to clone, you can click and you will get a complete recombinant DNA. After this, you should go here. You see this is a mark here, and here the yellow and green line. When you click here, you can easily see if your protein is expressing or not. So if you get a complete open reading frame for your inserted DNA, you can express in wet lab. So what happens after you designed, construct these steps, these are all computational. After this computational stage, you can order your recombinant DNA from GenScript and you can clone in E. coli. In E. coli, you can easily enhance the number of these plasmid, and then you can use mammalian cells, or you can directly use E. coli, but we prefer mammalian cells because it's easy to use. And you will have a high level of purity of your protein, high level of expression. That's why we use animal cells. After expression, you can purify using FPLC, HPLC, or column chromatography. And then you can use, like you have to check if the purity is good or not. You can perform some gel run. After that, those proteins can be used for identification of functional antibodies. So you have to select some plasma samples from children's or man or woman, and then you can use these proteins and the plasma to identify your designed vaccine can act as a functional vaccine or not. These are some references you can read. Thank you. Any question?
Ma'am, can you please go back to the slide? Um,
Which slide?
Ma'am, about your published papers.
So what you want, like reference? You want
Yes,
reference?
yes, ma'am.
Yes, ma'am.
Okay.
Okay, thank
Students,
you.
if you have any questions, you can ask.
Can we have the soft copy of your paper on the monkeypox? You said.
Yes, it's freely available. If you search
Okay,
on
ma'am.
PubMed, you can easily find.
Okay, thank you.
Ma'am, what kind of mammalian cells can be used?
So you can use HEC, or like I used HEC for my protein expression, HEC 293. Like it's a kidney cell, or you
Okay.
can use any other cell. There are lots of commercial cells also available. So whatever available in your lab, you can use.
They are from humans only?
Yes.
Okay, ma'am. So that culture is different in the labs.
Yes, it varies. But if you want to express in HEC 293, you just simply order and company will provide you the similar cell lines or similar type of cells.
Okay, ma'am. So suppose if it is, if the recombinant DNA is expressed, then we will have some kind of color change or any kind of antibiotic resistance in that. So depending on that, we will confirm that it is working.
No, no, no.
Hmm.
For HEC 293, we just express our designed vaccine candidate. So our vaccine candidate is a protein. So once you express your protein in this mammalian cell, you have to purify. After purification, you have to perform some ELISA. I hope you are familiar with ELISA.
Yes, ma'am.
Okay. So in ELISA, what we do, we coat our protein, our vaccine candidate, on ELISA plate, and we use human plasma. Like we know that human blood or human plasma has antibodies. So we take plasma from already infected. Suppose you are working on malaria. So we take plasma samples from malaria infected human and we use those plasma in our ELISA. So if that person has antibodies, that is going to bind our vaccine. This is first approach, okay? There are different approach you can validate your vaccine, but this is a very best approach, a direct approach that you can uh, validate your vaccine that, okay, this is my vaccine and the vaccine candidate has ability to induce antibodies or it can produce some functional antibodies by using those plasma. This is first approach. Second, you can use this protein, inject inside mouse and study the antibody proliferation. Like you have to collect blood every 3 weeks and you have to check the antibody level. Like if there is good titration of antibody or not. So this is second approach. So most of the time we just look at the antibody titration, but it's very raw. Your protein or your vaccine can generate antibody inside the mouse or human, but it should be functional. If the antibodies are not functional, all the high level of titration are useless. Before my postdoc, I was also thinking that, okay, if a vaccine is inducing antibody, it's a good vaccine. No. If your antibody is not functional, it's useless. It's not going further for vaccine development. So we always have to target functional antibodies. And that can only be identified by using plasma cells or plasma antibodies. Any other question?
Reverse vaccinology.
So reverse vaccinology is a computational approach. In that, we use the whole proteome of a pathogen. And after screening, like suppose there are 5,000 proteins in Plasmodium, we use different computational approach. And at the end of this, you see here, this is a reverse vaccinology. Here you see there is more than 208 proteins. And after that, you have only 8. These are different filters that provide different, like for example, vaccine is providing vaccinogenic protein, antigenic protein. Imagine provide the details of transmembrane helix. Signal P is providing signal peptides. So after every stage of filtering, at the end of this filtration, you will get highly antigenic proteins. So in wet lab, we use single protein or 2 or 3 proteins. But in computational, we are using the whole proteome. Okay?
Okay, ma'am.
Any other question? Okay, if there is no further question, I can start the demo. Okay, so first I will start with the Mala card. This is a database. Here you can enter the disease name. So here you can get all the details of this disease. And here, if you see, you can explore different details here, like what kind of drugs are and here you can also read about the clinical trials. Here you can study about the pathways. So here you can see there are lots of pathways that are targeting TNF, CD36. So CD36 also plays a very important role in immunity of this malaria. So what happens when Plasmodium interacts with CD36? It causes them to induce lots of complications. So we can generate some antibodies that can inhibit the interaction of Plasmodium and CD36. Here are some other proteins that you can explore. So these are all human proteins, host-based protein that are interacting with pathogen. So this is all for pathophysiology study. If you want to explore more, you can go to literature search. Let's start with SARS-CoV-2. I hope you all are familiar with this UniProt database. There are different options you can explore. If you go to the proteome and if you search, you can easily find there are lots of records here for SARS-CoV-2. And you see there are 17 entries. You can download from here, or you can direct click here on 17. You will get all the 17 details here. For more details, you can come to this side. On the left-hand side, if you see, there is 3D structures. There are 12 proteins of SARS-CoV-2 that are already showing. Like the tertiary structure is already available. If you click here, for example, replicase protein. Okay, let's go with spike glycoprotein. It's very common. So you see there is a term here, evidence at protein level. So whenever you are going to design a vaccine using UniProt database, always check if it's evidence at protein level or transcriptome level. This is another database you can explore. And this is the IEDB. This is the core of the vaccine designing. If you want to use already reported epitope or antigen, you can use this tool. But if you are interested for new or novel proteins, you can go use VaxiDB. This is a VaxiDB database, and it's also a vaccine designing tool. So I will come here later. First, I will show you experimentally reported antigens. So this is IDB database. Suppose you are interested in, can anyone suggest any disease or pathogen name? I will go with that. Suggest me any pathogen that you are interested to work on.
COVID.
COVID, okay. So it's SARS-CoV-2. We have organism here, and we have options that you can select T cell or B cell. So for now, I'm just showing all, like just all. You can also select human specific, mouse specific antigen. But here I'm just using any antigen. So here you can see there are 20 antigens. 20 antigens reported for SARS-CoV-2. So you can use any of these antigen for your vaccine development. So this is for SARS-CoV-2. So SARS-CoV-2 is a virus, so there is limited number of protein. What happens if it's Plasmodium? Because Plasmodium has more than 5,000 proteins. And if you're going to work on wet lab, it's very difficult to identify a novel antigen. It's very expensive. So for that purpose, you can use in silico approach or immunoinformatic approach.
Show the dengue also.
Okay, sir. So here dengue, there are different types. So what kind of virus affecting and what kind of virus you are interested, you have to select. Which strains, for example, type 3. If you are going to discover some vaccine for type 3, search. See, there is only 2 antigens reported for dengue virus. And if you come to the UniProt, you can search for whole proteome. See, they have only 1 or 2 proteins. So it's very easy to work on these kind of virus because they have very limited number of proteins. But if you explore the epitope, you can see there are more than 700 epitopes are already reported for this kind of antigen. And you can also explore the assay. You see the assay option here. There is T cell assay, B cell assay, and MHC ligand assay. So you can download this file from here. You see export result column at the right side. If you click here, you can see these kind of options and you can export. And again, you can also go for B cell assay. In B cell assay, you can also see that these are some mouse host and there could be some human host. But if you are interested specifically in human host, you can filter. So on the right side, you have filter options. Sorry, left side. On the left side, there is filter option. You see, if you click on human, and if you click on search, you see the number of epitopes decreased from 1700 to 1600. And essays also, the number is decreased. Now here you can see the host. It's Homo sapiens means human. And here you can also see the epitope type. You see the numbers. These are discontinuous B cell epitope. And here you see the linear B cell epitope. So if you want to study more, you can click here. Go to this. And you can see what are the details. So this is for experimentally reported. And if you download from here, so here you can see the PubMed ID, the epitope, And you can also see the assay method. So here they are using ELISA method. And they are using serum. And they are getting IgG. IgG is a kind of antibody. I hope you all are aware of this. So if you want to check what kind of antibodies they are giving, so most of the time there is IgG or IgM. Again, IgG could be Ig1 or Ig3. There are two different types. So yes, you can also read about these kind of details, like if this epitope is producing any kind of antibody or not. And the position detail you can also see. So you see here, this particular antigen can induce IgG. And if you see here, this particular epitope induces IgM. So there are lots of data on this website you can explore for experimentally reported epitopes. Come back to home. For prediction, you can use T cell epitope prediction. There is MHC class 1 tool here. For B cell, you can use this antigen sequence property. Like you can use Discotope for discontinuous and ELIPRO for full length. I will come back to this part. We will predict some epitope today. So this is another database. It's a VaxiDB database that you can use for identification of novel, or if you want to develop a vaccine for novel antigen, you can use this part. So here you can see organism type. If you are interested in virus, you can click on virus. If you're interested in bacterial, you can click on bacteria. So here I'm clicking virus. You can provide any proteome ID, or you can just select any virus from here. You see there is a huge list. Whatever you are interested, you can click and you can easily get the details. So
Mom,
here I'm selecting monkeypox. And you submit here. Yes, any question? You can
ma'am,
ask.
so
Yes.
immunity is localized or is it systemic?
Sorry, can you repeat?
Immunity, is it localized or systemic, ma'am?
In this case, like in what
Like,
context you are asking?
suppose if I want a vaccine that works like locally at lungs, particularly at lungs, suppose aspergillus, like aspergillosis is a condition where it is get infected with aspergillus niger. So suppose if I want to develop a vaccine against fungi and that like completely located to lungs, so only that part of the immunity does it work, or does
Yes,
it
yes.
affect whole body?
So the vaccine will work on whole body, but if you are interested on only particular lung region, you have to study what kind of immunity is going inside the lung. In case of SARS-CoV-2 also, SARS-CoV-2 is entering inside our lung, and what kind of immunity is going on inside the lung part. So it depends. That's why in the starting I told you we have to study about the pathophysiology of that particular pathogen. For example, aspergillus, there is different kind of pathophysiology, or the immune system working for that kind of aspergillus. So you have to focus on that part only. For example, if TLR2 is involved with this aspergillus, you have to work on TLR2 associated immune system. If TLR9 is not associated with aspergillus, what is the point to work on TLR9 kind of immune system? So you have to focus on TLR2 associated immune system if it's aspergillus.
Okay, ma'am.
Okay. Any other question? Okay. So here I just selected monkeypox, and this database provides us all this protein name, their antigenic score. So here, what we are doing in this stage, you see, I hope my PPT is visible to everyone.
Yes, ma'am.
Yes,
Okay, so here you have to perform everything, like you have to perform Vaxigen, TMHMM, Signal P, and then you will get these 8 proteins. But this database, VaxiDB, it's providing you all the score. You don't have to predict. It's already predicted for you. You just have to download this data. And here you can see the localization also. Like it's cell membrane protein or it's nucleus protein. You can see here. So here you can see the localization and also the probability. So the top, if you sort this list, the top are highly probable, and these are the proteome ID. So if you are interested to develop vaccine against monkeypox, you can use these highly probable proteins that are extracellular or it's cell membrane or it's endoplasmic reticulum. And if you see the signal peptides, these are signal peptides. They have 0 transmembrane helix or they have 1 transmembrane helix. And you see the antigenic score, it's also above the threshold. So we take threshold of 0.5. You can take 0.6 or 7 as per your research. So here you can easily see the name, extracellular, it's highly probable. So if you want to develop or design a vaccine against monkeypox, you can download and you can use. So now let's come back to the prediction part. So for prediction, we need a protein. So you can download the protein from here, or you can direct copy the sequence from this part. Here you can download from for sequence, text form or whatever you want. I just want text file, so I will take text file here. I hope everyone is aware with UniProt ID, NCBI SSN number. So here, when you come to the IDB database, you just have to click here. When you click here, you will get this kind of page and you have to paste your protein. And you can select the peptide length. You can predict 9, 10, 11 or 12 mer, whatever you are interested. You can also select like if you are using human alleles or mouse allele. So we are selected human. So this is the group of HLA that we are using for present prediction. And they're also providing the prediction method. You can use NetPen method, consensus method, ANN method. So I prefer consensus. And then you can play. It will start predicting for you. Similarly, for B cell epitope, you can use Discotope if you want to predict discontinuous epitope. You just have to provide the PDB ID. Just provide PDB ID. Okay. So this is the database where you can easily get the PDB IDs or tertiary structures. So 6V XX, this is the PDB ID for spike glycoprotein of SARS-CoV-2. You can use 6V XX directly and you can submit, or you can also select the chain. So here you can see it's trimer, ABC, it could be 3 chains. So that's why we are selecting any one chain. I'm selecting chain 1 and you can submit. It will take some time. Meanwhile, we can check the result for this. So here you see, these are the predicted peptides, the start and end position, the peptide length and the length. You can download from here. So once you download, you can see the details of alleles here. These are different alleles for different peptides with different scope. You see, there are lots of predicted epitopes. More than 91,000 epitopes are predicted. So we can't use 9,000 epitopes for our vaccine development. So what we can do, we can use the percentile cutoff. In this case, if you take 0.5 as a cutoff, or you can use 1. Like 1 is a good threshold for this. Yes, up to 1. You can select up to here. Now copy all these. So why I copied in the next page? Because these are good, like these peptides or epitopes has good percentile rank. And if you are going to sort this, what happened? You see, there are duplicate epitopes, but these are not duplicate. If you see the result, the single epitope, this epitope has 2 alleles. Similarly, if you see here, the same epitope has 3 different alleles. So what you can do, you can just copy and press tab. And you can copy the alleles, whatever alleles are for the same epitope, you can paste here and make a list where your epitope based alleles are in a comma separated form. This kind of file. Once you prepare this kind of file for all your epitopes, here there are lots of epitopes, duplicate epitopes, but with different alleles. You see, this epitope has HLA-B and HLA-A both. Once you prepare this kind of list, you have to perform PPC. PPC stands for Prediction of Population Coverage. You see here, Population Coverage tool. Come to this tool, and here you can use world analysis, or you can select India levels analysis. And you can choose the file or you can paste. So here we prepared only one for now. So you just have to paste your epitope. Yes. If you have, once you prepare whole list, you can upload from here. There is upload option. You just select on choose file and you can select. So what happens when you enter your epitope and ellipse? You just have to select the country. So if you want to run this on world as well as on any other country, you have to press control and select. If you want to select more, just press control and then select. And then you have to submit. Okay, you have to select class 1 also. You see, the same epitope that has 26% coverage in Europe, but in India, it's only 7% of coverage. But if you look at the world, it's showing 20% coverage. And this is only for single epitope. Once you have a group of 10 epitopes, this will increase up to 99%. So for that, you have to prepare a list of epitopes with their respective alleles, and then you have to run. So you can clearly see the difference that in Europe, its coverage is 26, and in India, it's only 7. Here you can see, and you can download this result also. So we got the discotope result also. This is the B cell epitope prediction that we are performed. So the green region is good epitopes. If you want to see in table view, click here. So these are the predicted epitopes. If you want to see in 3D, click on 3D view. You can see this 3D view. The yellow region, here you see the yellow, all the yellow colors. These are the epitopes. You see, every time you click, there is a red ball here. You see the red balls, these are the predicted epitopes. And all are on surface. So epitopes should be surface exposed. If it's embedded inside, it's useless. Don't use the epitopes that are embedded inside the protein because it will not be available to our immune system. It's useless. Always use the epitopes that are surface exposed. So once you design your vaccine, go to the PyMol and check what are the epitopes that are available on the surface. And you can write those things in your discussion part of your publication. Okay, so this is the epitope prediction part. After prediction, we will go for vaccine design. So here in this slide, I have told you that you should design your vaccine in different combination of epitopes so that you can get a best version of your vaccine. For that, you can use the VacciDB software. Here you can see the design option. Once you click on the design option, you see vaccine design page. And here you can write your vaccine name and you have option to select adjuvants. You can select adjuvant linker. And for adjuvant, you have options like you can use colotoxin B, heparin binding. These are already reported adjuvants, but you have another sequence, like if you have some other sequence, you can also select other. But here I'm selecting, this is a very popular adjuvant. And if you have predicted some epitope, you can write here. For example, I'm just typing a random epitope. Suppose you predicted this B cell epitope, and you also predicted some T cell epitope. So we have to, okay, so before going for T cell, we have to also select some linkers. For B cell epitope, you can select this or any other linker. So right now we have T cell epitopes. So I'm taking these two T cell epitopes, and you can paste. So we predicted CD4, so I'm typing CD4 here. If you predict CD8 T cell epitopes, you can also use CD8. For now, we have only CD4, so I'm just pasting here. Now you select your linker, and whenever we design our vaccine, we have to add his tag. So what is his tag? His tag is a continuation of histidine that helps you to locate or to identify that if your protein is expressed or not. So this is a kind of marker that will help you to identify or to locate if your vaccine is expressed or not. So here you can also select the order, like if you want B cell first or T cell first. So I'm selecting B cell first and then CD4. And if you want vice versa, you can select CD4 first and B cell again. Like whatever you want to design, and just click on generate. So here you can see different combinations of the same epitope. You see the green part, this is the linker of adjuvant. The red part, this is adjuvant. And this blue region, this is your epitope. And this yellow part, this is his region. So here you have total 30 combinations. You can download or you can copy paste from here, and you can predict the tertiary structure. Once you get your tertiary structure, visualize on PyMol. If you get good surface exposed epitopes, suppose you have got good surface exposed epitopes on combination 2, then only go for molecular docking using this sequence only. If this combination 1 is not showing good surface exposed epitopes, don't use this combination. Now we have protein sequences. You can use ITACR for prediction of your protein structure, tertiary structure. Then is next remaining. Okay. For docking, I hope you will have some classes for docking. And for vaccine expression, you can use SnapGene. Any question for now? It's very simple to design vaccines. It will take only 5 to 6 days to design, 5 days for docking and simulation. Simulation may take long time because it's protein, so it will take more time. Any questions? Okay, if you don't have any question, I will continue. So for continuous B cell epitope, you can use EryPro. And if you submit here, you have to select the chain. Here you see the continuous sequence. This is the linear B cell epitope. And if you want to visualize it, you can visualize here. The yellow ball, this is the selected B cell epitope region. You change You can see these are the linear B cell epitope. So similarly, you can use some other tools that is already listed in this. Here, there are lots of tools that you can use. Okay, what else? So after molecular docking and okay, it's not working, I think.
Select advanced. Click on advanced.
Sir, this will not work here right
Move.
now.
नहीं, नीचे नीचे जाओ. Proceed to www. Last में. हाँ, इस पे क्लिक कर लो.
Okay, so this is another software that you can use, CADD. So here you just have to paste your sequence. So here you just have to copy this vaccine combination. So this is a protein. Remember, this is protein, and we are going to convert this protein into DNA. So I just pasted your vaccine sequence here. And we are going to convert this into DNA sequence, step 2. Now we have to select the organism. So which organism we have to select? Can anyone suggest which organism we have to select here?
Can we select, ma'am, Nipah virus?
No. We are going to express our protein. So we need
Okay.
expression body. Like we express our protein E. coli. So I'm selecting
Okay.
E. coli.
Okay.
If you are going to express your protein in human cells, you can select human. But here I'm choosing E. coli because we are mostly using E. coli in our lab. And then we can select avoid these things and we can submit. Okay. We have to select, we pasted protein sequence. So we have to mention it. Okay, now you can see improved DNA here. And these are the DNA sequences. The CAI value is important here and the GC content is important. So GC content always considered as good if it's more than 50. And CI value if it's more than 90, 0.95 or 0.90. And you see the translated form here. I hope you all are aware with codon things. Like in bacteria, different codons translates different amino acids, and in humans, different codon translates different amino acids. So you have to check if it's same or not. your protein sequence. And here what you have to do, you have to copy this cDNA and prepare a separate file. And this is going to upload on here, SnapGene. On SnapGene, here is option of vector. When you click on vector, you have to upload from here. Okay, wait. Okay, so here you see the vector. When you click on vector, you can select the options from here. And when you click on insert, you have to upload your cDNA that you just copied from that software CADD. Once you upload here, you can get these kind of options. And you will have some kind of restriction enzyme options also. So what you have to do, suppose you want to target this BAM-H1 and XBA1, but your protein don't have that restricted sites. I hope you all are aware with that restriction enzyme and recombinant DNA technology. If not, you can study or you can ask me anytime if you are interested to develop this recombinant structure. What you can do, you just have to add some like flanking regions from this part of this region and this part, this region. How? I will show you. For example, this is your, okay, this is protein sequence. This is your DNA sequence. Just copy all the sequence. We all know that every restriction enzyme has some specific sites, restricted sites. So what you have to do, you just have to add restriction site at your NNC terminal. For example, there is a restriction site for CAG. So this is the restriction site for BAM-H1. So you just paste here. And this is the restriction site for XBA1. Okay. So what happens once you add those restricted sites on your N terminal and C terminal of your vaccine, this software is going to take that as a restricted site. But beside this, what you have to do, you also have to add one start codon. You know, can anyone tell me the start codon? What is the
From
start?
where the application start means recognition site.
Yes. So we all know that AUG is a universal start codon. So after entering your restriction site here, you have to paste start codon so that your plasmid can identify that, okay, your plasmid system can identify, okay, this is the restriction site. We have to cut from here, but we have to start our translation from here and it will start till the end. Can anyone tell me stop codon? For example, this is a stop codon. So you have to add stop codon at the end of your protein sequence. So this is not protein sequence, this is DNA sequence. So this is stop codon at the end of your cDNA. Okay, so this is going to be your insert fragment. So every time you are ready with your protein, translate into cDNA, add, this is your restriction site, add your restriction site, then add your start codon, then your cDNA sequence, and this cDNA will end with the stop codon. Like you are giving a off signal that you have to stop the protein synthesis.
উঠে নিয়েছিস?
and then the restriction site for second enzyme. Now this is ready to upload your file. Once you upload this file, it's a guarantee that you will get this complete recombinant structure. It's a guarantee. Okay, so if anyone getting any difficulty for designing your recombinant protein or DNA, recombinant DNA, you can ask me. This is my email ID. If you have any problem in future, you can ask me. For now, any question?